Good evening. I'll ask everyone to take their chairs, take these white chairs, please. In the blessing of God's good creation, including this cool, refreshing, much longed for rain, welcome to Canadian Mennonite University. You know, I think there's a thing in Winnipeg, doesn't matter what the snowstorm is, people will go to everything, but if there's rain, it's somehow reason to stay home. There's some habits maybe that we can work on with others. In any case, my name is Cheryl Pauls and I serve as president for CMU. Whether you're here face-to-face in person or face-to-face online, it's a joy to be together, to be provoked, to get dirty, to get the lowdown on Earth's fabric in our own yards, to get inspired as stewards of community yards, backyards, and front yards, and what Manitoba's Minister of Environment and Climate Change, Tracy Schmidt, calls fjordens. Think about that for a moment, fjordens, and where that's putting us. We gather here together in the presence of a God of extravagant mercy at a time when we are tuned to the fragility of so many who are hurting and weary, the fragility of the land and life itself. At this time and always, may our longings, our learnings, our laughter be acts of hope and healing. At this time and always, CMU is honored to live and learn on ancestral lands and in the waters of Anishinaabe, Cree, Dakota First Peoples, and the homeland of the Red River Métis Nation. In seeking justice and the flourishing of all, we honor the gifts of Treaty 1 and these Treaty 1 plains as covenanted before Creator in 1871, the cool, clear drinking water of Treaty 3, and the hydroelectric power of Treaty 5. To pursue gifts of healing and hope in the soil, I'm pleased to introduce this evening's host, Evan Balzer. Evan is a CMU alumnus with a Bachelor of Science in Biology from University of Manitoba, a Master in Science in Biology from University of Waterloo. Currently, he is working as project coordinator for Nature Conservancy of Canada. And Evan will introduce tonight's guest panelists, and I think he'll let us in on a bit of what he thinks too. Welcome, Evan.
Well, thanks so much for allowing me to join you today. I spend a lot of time in my work connecting with folks from a variety of backgrounds, and so I'm very excited to be sharing the stage with you. And coming back, you know, I was just speaking to my partner. We hadn't been in here for a meaningful length of time in quite a while, so it's good to be back on a stage with you folks today. One of my all-time favorite movies is the Disney cult classic Atlantis: The Lost Empire, whose heavy-handed critique of colonial imperialism would only be appreciated later in my life, but whose imaginative machinery and eccentric cast of characters captivated my young mind. One of whom, a specialist nicknamed the Mole, becomes incensed when the movie's protagonist bumps the mole's collection of international dirt samples, stating, "You have disturbed the dirt." What first struck me as a throwaway line, quotable anytime someone knocks over a sandcastle or mucks with a carefully organized garden plot, has taken on new meaning for me as I have worked more and more in prairie ecology. Winnipeg and the region surrounding it is near the northern limit of the historical tallgrass prairie, which once extended from as far south as Texas to the Interlake region here in Manitoba. Tallgrass prairie is characterized by charming perennial grasses and wildflowers, whose deep root systems allow them to persist through freezing winters, hungry bison, and cyclical wildfires. After thousands of years, one legacy of this ecosystem and the indigenous communities that stewarded it is rich, dark soil packed with nutrients. Upon settlement, it became apparent that this rich, dark soil was in turn excellent for agriculture. And within only a few hundred years, well over 90% of the tallgrass prairie ecosystem here in Manitoba was converted for other uses. Today, the species that rely on this ecosystem are in steep decline, and our connection to that soil, the plants that give it life, and the smell of it on our fingers are increasingly distant. You have disturbed the dirt. It can be easy to dwell on that part of the history, on the loss and the change that surrounds us. But in that loss and in that noise, I think it's important to step aside and to reconsider our place within that story and to recognize our interdependence with that soil itself. From ranchers using sustainable grazing techniques to indigenous community leaders sharing knowledge about traditional medicine harvest, my work has brought me into spaces where the health of the land and its soil are among the primary concerns of its stewards. Indeed, there are countless ways to examine dirt, and I am pleased today to share the stage with some of the finest folks to do so. These folks beside me have investigated community-led climate resilience, the tiny dramas that play out on soil's microscopic stage, and the ways in which food production can go hand in hand with a healthy earth. So today I'm happy to introduce you to the following people. Heather beside me here, Heather Unger and her husband, Paul Krause, own and operate Northern Grove, a business that revolves around restoring healthy relationships with earth through tree care, permaculture design, and soil health consultation. Rather than seeing human activity as necessarily damaging to the earth, Heather and Paul seek to partner with it to benefit not only through regeneration of ecosystems, but also to create ecological and economic resilience and to benefit their community as well. In that sense, they hope to make as large a footprint as possible. She today will give us a rundown on what exactly soil is, because it's a great question, and also how when we understand the fundamental realities of soil, we can build more sustainable places upon it. Next, Dr. Rachel Krause, an associate professor of biology here at CMU. Rachel's teaching and research spans ecology, parasitology, and global health, taking her from the icy streams of BC to the mighty St. Lawrence River in Quebec, to a research partnership with the University of Panama, and across the pond to an agricultural project in Zimbabwe. Today she'll be sharing about that work in Zimbabwe in partnership with local communities as they implement innovative land-based climate adaptation strategies, a big phrase we'll get into a lot later. And lastly, Dr. John Brubacher, Associate Professor of Biology here at CMU. Now, one of John's main research topics is using flatworms to understand a particular kind of cell called a germ cell, which is important in the production of sperm and egg cells. And when he isn't publishing work on flatworm photography, you can ask him about that later, he's exploring how soil microbes can inform our battle against antibiotic-resistant disease, and that's what he'll be talking about here today. And lastly, you, the audience today, have a role to play as well. I'll be inviting you to ask questions of our panel later in the evening, and I encourage you throughout to be curious and attentive. Be a 3-year-old, shovel in hand, helping mom in the garden for the first time. Be teenage you, getting dragged along to a farmer's market and tasting soil on a freshly picked carrot. Author J.B. McKinnon, whom I love oh so much, says that there is much to be gained and nothing to be lost in such small acts of reconnection, and I tend to agree. And so with that, friends, Heather Unger.
Thanks, Evan. That was a great start. So as Evan mentioned, my husband and I started our business, Northern Grove, with a focus on healing human relationships with the earth. And we've done that through our work in tree care, in permaculture design, and consultation. And then upcoming in spring, we'll be launching into soil health consultation. Just for a bit of background, our work is based on I need to be flipping this. So that's our business. Our work is based on a few foundational beliefs. Namely, we believe that we are made in God's image to co-create with God. We believe that human presence on earth can be healing. We believe humans can adjust the way we live to align with the way earth works. And we believe that we, humanity, can make dramatic changes to make this happen. and to showcase what's possible. So these are the beliefs that undergird our work. These beliefs are actually also the result of a lot of inner work that Paul and I have done over the past number of years to move us through ecological grief that we share in common with so many, so that we can move through our ecological grief and despair to a position of hope and action. And the reason I share this is that people are looking for models. One of the most common questions we get is, I'm freaking out, what can I do? And my response is this: we first need to do whatever inner work it takes to move us through despair. Once we have done that, we can assume a posture of composure and hope. And then when the appropriate action becomes clear, we're in a really sturdy and grounded position to act. When we started our business, I contemplated this phrase: imagine a world where life springs up underfoot wherever we tread. Well, as it turns out, we found a way. Soil health regeneration is one area where we can quite literally bring this about and see demonstrable results in a very short amount of time. So today I'd like to share a little bit about how soil works, and then share real-life examples of how Earth responds when we bring soil back to life. I'm relying heavily for my presentation on the work of Dr. Elaine Ingham and soil food web consultants Ronald Flores from Peru and Todd Harrington in the US. In 400 BC, Plato wrote, "The land was the best in the world, and in comparison of what was, there remains only bones of the wasted body." In 400 BC, Plato was describing soil depletion. So what could have caused soil depletion back then in his time? They had no large-scale machinery, they had no chemicals, but whatever they were doing still led to desertification over time. Two things: tilling kills soil microbiology. We dig it, we churn it, that impacts the life that's in the soil. And two, an inadequate return of organic matter to the soil, which is actually the food that soil life requires. So we kill off the soil biology, and we remove soil life's food source, and slowly we will observe a decrease in soil life over time. In terms of overgrazing, we're not churning up the soil, but we are removing plant matter, which does a couple of things. Again, it removes the organic matter that soil life relies on, but it also shuts down photosynthesis, which is a driver of activity in the soil. Overgrazing also leads to compaction, which creates an anaerobic environment, which is not hospitable to the soil life we're looking for. So if soil is depleted through killing off soil biology, removing organic matter, and creating anaerobic conditions, what does good soil look like? Well, kind of the opposite. First of all, we need a mineral base, right? The mineral component to the soil that provides the nutrients and the micronutrients that plants need, or that soil life will extract and benefits plants. So that's the sulfur, the potassium, the calcium, and so on, the molybdenum, all the little micronutrients. So mineral component. Then we need organic matter, again, to feed the soil life. And we need the existence, the presence of aerobic soil life. I won't get into this slide in too much detail, but here's a visual put together by Dr. Elaine Ingham and just to give a little bit of background on Dr. Elaine Ingham, she has been a front runner of soil health study. She's got 40 years of experience in the field and has been training people up in how to regenerate soil, what she calls the soil food web. So here we have a visual of the soil food web of aerobic soil life. So in the soil food web, we have 4 major players. We have bacteria, fungus, nematodes, and arthropods. And just like the food web above ground, we have a cycle underground of predator and prey. And if one of those players is missing, the food web is thrown out of balance or collapses. So what does soil life do? A number of things. First of all, soil life builds soil structure. It creates what we could call a soil sponge. Here, the bacteria and the fungi glue together little bits of organic matter and the sand, silt, and clay particles to form aggregates, microaggregates and then macroaggregates. The effect of this is that just like a sponge, the soil is held together and doesn't wash away. But also, the effect is that passageways are created, which allows water and air and roots to pass through easily. Pockets are created, which allows the soil sponge to retain water and nutrients as well. And finally, the soil sponge acts as a filter, so that when water passes through it, it passes through and is cleaned. And when it exits the soil sponge, it will exit cleaned. I'll share 2 slides of demonstrations of how the soil sponge works, put together by a woman named Dee Pershouse. She's someone to look into. First of all, here's a demonstration of 2 plates. One has a pile of flour, one has a piece of bread. On the first pile, we see with the pile of flour, the act of pouring water over that pile of flour demonstrates what happens when rainfall falls on exposed soil, on tilled soil. And you can see the flood drought effect at work, where water pours down on the top but runs quickly off, and the top of the pile is in drought conditions, while in the lower parts of the plate we see flood conditions. We also see that the water has taken along sediment with it, so the water is quite muddied, right? And we see this effect in springtime when water is washing off of tilled fields, right? Our ditches are filled with muddy water. In contrast, on the right, we see a piece of bread demonstrating how flour or soil is held together when we add biology. The bread, or the soil sponge, first soaks up water, retains it, distributes it within the sponge, and then any excess water comes out clean. A second demonstration showing a progression from an intact soil sponge on the left to a completely depleted soil with no soil structure on the right. Notice on the left how little water, if any, comes through. Most, if not all, of the water is retained by that soil sponge. And as we progress to the right with increasingly depleted soil, look to see just how much, first of all, how much water passes through. The water is not being retained by the soil, and how much sediment is brought along with the water. So, soil life creates a soil sponge, and that's significant, particularly in our context, in our agricultural context, where we are increasingly seeing flood drought dynamics at play, and how the soil sponge, how soil structure can actually temper those extremes. What else does soil life do? Well, it enters into a collaborative relationship with plants. Studies have shown that every soil, the world over, sand, silt, clay, pebbles, boulders, every soil type has every nutrient and micronutrient that plants need to thrive. The catch is that plants cannot, on their own, extract nutrients from that parent material, from the sand, silt, clay, and so on. Plants require a helper. Plants require soil biology to extract the nutrients from that parent material, the sand, silt, and clay. The soil biology being the bacteria and the fungi, which extract the nutrients and then that concentrate those nutrients in their being, in their bodies. And then as the nutrients are cycled through the food web, through the predator-prey dynamic, those nutrients are converted into a plant-available form that plants can finally take up and consume. And so what the plants do is they will flush sugars out into the soil to attract the soil biology, the bacteria and the fungi, to the root zone. And the bacteria and fungi will consume that sugar, and it becomes this exchange of sugar for nutrients because of all of the activity among the soil life right around the root zone of extracting nutrients from the parent material and converting it into a plant-available form. So, soil life cooperates with plants. A few more functions of soil life that I'll go through quickly. Soil life protects plants from pests and disease by coating, actually physically coating the plants from root to tip with themselves and creating a protective layer that protects plants from pests and disease. Soil life suppresses weeds in soil by shifting the bacteria to fungus ratio in the soil away from a bacterial environment, which favors weed growth, toward a more fungal environment, which favors weeds less and less as you go along, toward a more fungal environment. Soil life sequesters carbon. As fungal strands grow, they store carbon within their strands, within the fungal hyphae. And the carbon storage potential of fungus, of these fungal strands, is tremendous. There is an example that I'll give of a fungus in Oregon that is estimated to be between 2,000 and 8,000 years old, and is estimated to be the size of over 1,600 football fields in size. Imagine the carbon holding capacity of that fungus. And finally, soil life decomposes toxins. It takes the toxins that come into the soil, breaks them apart, and stores those little parts within the soil structure. So how do we build soil health? Well, in fact, we can reintroduce biology into depleted soil, almost like a probiotic. I'll take you through some examples of what happens when we reintroduce or inoculate depleted soil with a balanced soil food web. The following slides are test plots that were run by a company in the early 2000s in Idaho. They ran trials of across over 25,000 acres of production. On these test sites, they applied soil biology in solution, meaning in a applied soil biology in solution so that it's easily sprayed onto fields, to fields of alfalfa, potatoes, beans, corn, and peas. And the results were remarkable. Across the board, higher yields, more resilient plants, and lower costs to the farmer. So here is an example. This first picture is of a sugar beet field. The farmer, with one application of soil biology, this farmer experienced an increase in yield of 42% with a higher sugar content in the sugar beets. Here, alfalfa fields, where they were experiencing heavy compaction in some areas, stunted growth in their alfalfa, and space between their rows. So really not a thriving crop. With one application of soil biology, they observed decompaction in previously compacted areas and an increase in yield of 4 tons per acre in 3 cuttings, so 3 cuttings over the course of the summer, to 7 tons per acre in those same 3 cuttings. Here we have pastures run by an organic dairy. They had been previously, the pastures were stressed because of previous management practices. But with application of soil biology, they observed higher feed value and an increased protein content in the grass from 4% to 6%. That's an increase in protein content of 50% in one application. Same farm, maybe different fields, but again, after applying soil biology, not only did they observe an increase in protein and health in their pastures, they also observed weight gain among cattle, improving by 25%, and a reduce in weed pressure. The only complaint that this farmer had was that he had excess grass that he had to bale due to increased yields. Potatoes. Here, this farmer had problem with low yield and potato beetles. After an application of soil biology, this farmer experienced an increase in yield of 42% and an elimination of the problem he had with the Colorado potato beetle. I don't have a slide for it, but they also ran trials on peas, where they experienced an increase in yield of 136%, with decreased costs to the farmer by moving to a biological approach or biological inputs. Here's a fascinating study run by a different company in Sweden. These were actually studies run by the gentleman that I mentioned, Ronald Flores. He's in South America, but these tests were run in Sweden, where this particular organic market garden was experiencing pest problems and weeds. They had brought in commercial compost, and so they ran 3 trial plots among their various crops. So they had a control. The scenario 1 is their control with no inputs. Scenario 2, application of commercial compost. Scenario 3, with the commercial compost as well as an inoculation of soil biology. Fascinating thing was that with the application of the commercial compost almost across the board, in that scenario, that second scenario with just the application of commercial compost, they experienced a decrease in yield and an increase in yield pressure. So it's really important, that highlights the importance of making sure that your compost is a good compost and has this microbiology in it to do the work. In contrast, the third scenario with the commercial compost and the microbiology inoculation, overall decrease in weeds of 38% and an increase in yield of 72% across 8 different types of crops. And I'll go through these slides quickly because the findings were generally the same. So in their celery crop, with the inoculation of microbiology, they observed a yield increase of 149%. Fennel, increased yield of 58%. Onions, yield increase of 12%. Potatoes, yield increase of 42%. The one critique of this particular plot was that they're not the same variety. In their defense, they were all high-yielding varieties, and so take it or leave it. But the observation they had was that they had an overall yield increase of 42% with the microbiology inoculation. Rutabaga, 73% increase. Lettuces, 87%, and Swiss chard, 63%. So of course these examples are focused on an agricultural context, but really the principles can be applied in any context involving depleted soil, whether it's orchards or clear cuts or deserts or forests or urban environments. The principles are the same, that where we have depleted soil, the key is microbiology, organic matter, and aerobic conditions. So just to wrap up then, what can we do? Well, as it relates to soil, we can learn about the soil food web and what makes good soil, the mineral component, organic matter, aerobic organisms. And then we can take on practices that are amenable or that support soil life. Shift to no-till. Cover the soil. Feed the soil. We can take on a model of permanent beds. Use perennial covers. I know even large-scale farmers are taking on that approach, that they'll seed their fields to a low-lying perennial cover and then seed into that for their annual cropping. Mulch, organic matter, and then of course, where opportunity exists, we can use inoculations of soil biology, which is what Paul and I are planning to launch into this spring. And finally, I'll end with this, that I think that's my cue. I don't know. It's okay, I'm almost done. And then finally, I would suggest we can do that inner work that I mentioned at the front end. The process of moving through despair and grief is important. And it's not something that we can go around. We go through it. But we can and surely can move through it with intention and come to a place where we are composed and can move forward with hope and action. So thank you very much for your time. I look forward to any questions you have. I'll leave this here.
So as far as format goes here, I think I'll just throw one quick question to you, Heather, and then we'll kind of do, you'll speak, and then I'll provide a question, same thing through, and then we'll do some responses. So I think one of the things that I wanted to quickly touch base on before we moved along was just when you think about, so someone who's got like a garden box at an apartment that they're renting, or a backyard space at a home or a community garden nearby, like what are 2 or 3 practical things that you see us missing, or opportunities that we can jump on to put those things into work? And I'll give you 2 minutes, just the highlight reel here, and then we'll move along to Rachel.
Right, I think, well, a couple of things that we can do right away is make sure that our soil is covered. No need to till. When we don't till, we actually nurture an aerobic growing environment. And then if we keep it covered, we keep our soil biology healthy. So maintain aerobic conditions and then feed that soil biology with mulching and leaving organic matter on the soil surface. As the last example suggested, compost can be great. It can also be really bad. So just a little red flag on the compost one. Yes, compost is a great idea if you have a really good source.
Awesome. Thank you so much.
You're welcome.
Yeah, oh yeah.
All right, so we've heard a lot about soil at home, soil where we live, and I think that's often what we conjure in our minds with soil, right? Soil is the material under our feet. It's immediate and local, And you alluded to this a little bit, but it tells the history of the place, right? What's the underlying parent material? What has happened in this place over time? The soil really tells that story. I would argue that soil also connects us globally. This is partly because soil is doing all of this cycling work, and we heard about this already, kind of. There are lots of things that cycle through soil and the atmosphere and around the world in this way. Water, nitrogen, sulfur, maybe these are elements, and materials you've heard about. And in reality, actually, they all cycle together, and they influence each other, and importantly, they cycle along with carbon. So I'm going to focus on the soil, climate change, carbon dioxide connections here as well. So this connection globally through the element of carbon happens through opposing processes of photosynthesis and decomposition, right? Carbon is taken in the form of carbon dioxide by plants, turned into sugars, ultimately into the plant's own biomass, right? Into wood, into solid mass. And then as the biomass dies off, drops to the ground, undergoes decomposition, returns that carbon back to the atmosphere. And I think when we think about the food chains that we heard about as children, we get this really nice picture of, you've got the grass, and then you have the bunny come and eat the grass, and then the fox comes and eats the bunny. But in fact, the vast majority of what is eating that plant material isn't those cute herbivores, at least not animals eating the plant while it's alive. Most plants aren't consumed until after they've died and returned to the soil, in fact. And it's that interface with the soil that's key in the whole story of climate change and carbon and soil. Because that's where most of the life that eats plants, or dead organic matter, as we would then call it in ecology, that's where those organisms hang out, right? We heard about the soil invertebrates and the fungi and the microbes and nematodes, maybe you noticed that word. They complete that photosynthesis decomposition cycle and return that organic carbon back to the atmosphere. And it's through these processes that the soil that we're locally standing on connects us to the global world through climate. And therefore, when we're thinking about adapting to climate change, as we are in this project that I'll tell you about in a moment, we need to think really seriously about soil, not only here at home in Canada, but also around the world. So I'm going to tell you a little bit about LINKS now, locally led indigenous nature-based solutions for climate change adaptation in Zimbabwe. So you can see why we have an acronym here for it. The truth is that many of us, all of us, are connected to the soil in some way, and many people's livelihoods are directly connected to the soil, right? I'm talking about farmers mostly. And LINKS is working with farming families to implement what we're calling nature-based solutions. So you can see that term in that title. So this is a term that we're using these days. It's got political currency. This is a Global Affairs Canada large grant project. So we're using that language. But the ideas and practices that it encompasses are by no means new. LINKS recognizes this by answering this new terminology, nature-based solutions, with traditional knowledge systems, indigenous practices along with those. So what do we mean by nature-based solutions? Actions that protect, sustainably manage, and restore natural and modified landscapes that are also trying to address societal changes effectively and adaptively, and also provide benefits for humans as well as biodiversity. So it's really trying to do everything, right? We're trying to protect the planet, we're trying to care for people, we're trying to protect other diverse life forms. Often we're talking about agricultural types of nature-based solutions in this project. So these are long-term, cost-effective. Here we're trying to tackle sustainable land, water resource management, and climate change, all together. So these are practices that improve water availability and water quality, and also restore ecosystems and soils. And many of the nature-based solutions that are being applied by links are also practices found in indigenous knowledge systems and are related to caring for the soil. So things like intercropping in particular ways. In North America, sometimes we talk about the three sisters, right? The corn or the maize and the bean that climbs up the corn stalk, and the squash with the wide leaves that protects and covers the soil. And in different indigenous settings, there are other similar kinds of traditional partnerings, intercroppings. This is all great, but these practices, these indigenous practices, can fall apart under social and environmental stress. So nature-based solutions are trying to shore those up and add other evidence-based methods to complement them. So let me give you a few examples here, what we're talking about. And I'll show you a few photos from the field. These are mostly photos that I've taken, and you'll see me in a few and colleagues in Zimbabwe in a few. So alongside Mennonite Central Committee and Mennonite Central Committee Zimbabwe's on-the-ground partners in different districts in Zimbabwe, a variety of different nature-based solutions are being supported. Some of these have been in place for quite a few years in lots of communities, and some of them are new or they're expanding into new communities. The big one is conservation agriculture. And these days we like to talk about it as conservation agriculture plus, because we're recognizing that it's not just that households need to be able to grow food sustainably for their own usage, but they also need to be connected to markets, right? So thinking about the whole livelihood basket there. Conservation agriculture, you can see some examples here. So on the left photo, you can see a field that is just being set up, and you can see that the farmer here has set out plots that she's going to be planting in. In the middle photo, you can see my colleague, Dr. Luke Jimu. He's doing some soil sampling here, and you can see kind of in the bottom left of that photo, you can see, I think it's a squash plant, and you can see some maize and some other things there. Conservation agriculture is a production system that is caring for soil through managing the various elements of the farm, right? Looking at crops, soil, nutrients, pest management, water, energy management as well. All of these managed based on sustainable ecological foundations provided by three interlinking principles. So thinking about minimum soil disturbance, where have we heard that? Right? No-till, direct seeding, for example. Maintaining soil cover, so mulches, crop residues left on fields, cover crops. I'm just repeating things that Heather said now. And diversification of crops. Rotating crops through, looking at different traditional associations, and also looking at cover crops. So it's not an accident that you're hearing a lot of repetition with what Heather has talked about. These are globally useful management techniques. You can see on the far right here, Luke in the top, Dr. Juma at the top right, setting up a pitfall trap, and at the bottom you can see one that's been set in the ground. Maybe you notice that there's kind of a green bubbly liquid at the bottom of it. So we use this sort of simple technology of plastic cups and detergent to collect arthropods walking around on the soil to see what kind of arthropod diversity is coexisting in these conservation agriculture plots. Because with lynx, we're not just concerned with farming and farmers' livelihoods, but we're also concerned with biodiversity, right? We're concerned with conserving the rich varieties of life that we coexist with on the planet. In ecology, one of the general patterns that we observe is this idea of diversity begetting diversity. If you have more diversity, you'll have more yet. It's sort of a truism, but you can imagine, right? You have more different kinds of plants, that you automatically have more ways for other things to live in and on those plants, right? We would call those niches, if we're going to use really technical terms, right? The more diversity you have, the more niches you have, the more different lifestyle opportunities, maybe we could say. And so then the more life you will see inhabiting those areas. And this is certainly what we observe. So this is some preliminary data. We just got this back in the last few weeks. So this is late breaking, hot off the presses. You can see on the left-hand side and the right-hand side, maybe you can see some differences there already, right? So I'm not gonna ask you to guess which is from which. Maybe you would, hopefully you would guess that the left is from conservation agriculture fields. And you can see there's all sorts of different things there, right? So the orders, so high taxonomic level is in the caption there. So there's lots of beetles, those are the coleopterans. There's some flies. Orthopterans are different kinds of crickets and grasshoppers. Hymenoptera are bees and wasps and ants. Homoptera, other kinds of bugs. Lepidoptera, which are caterpillars and moths and butterflies. And then you've got spiders as well. And we've got one giant millipede at the bottom there. So just an immense diversity of things. And that's really, like, we would lift these cups out of where they were sitting in the ground, and you would just see it in there. The cup was full of all sorts of weird and wonderful things, right? So this is just from one district. This isn't even everything we saw. Right-hand side, there's still some diversity there. What we're not seeing there is that most of those things in that cup that we pulled out were just ants. You'd pull up the cup, and maybe you'd have a few spiders or a few ground beetles, and then you just have ants and ants and ants. That's what was going on in these fields, so in these more conventionally farmed fields. And these fields are like 10 meters apart, right, these samples. It's not like we went way over here and then we went way over here. They're right next to each other, that we're seeing this kind of dramatic difference in biodiversity. In the regions where lynx is operating, we're concerned with soil and soil fertility because of the agricultural livelihoods of people. But there are other more alarming soil issues that we observed as well, that people are living with. Soil erosion resulting in gullies. And you can see on the left here the beginning of one. We're kind of standing on the bank there. That right inset is quite a deep one. It was maybe 10 feet deep. I'm gonna mix my metric system here, but maybe 20 meters from someone's yard, right? And there were others close to schools. These are alarming signs of climate change and of ecosystem degradation because of changing climate and other pressures. So part of what Lynx is doing as well is looking at ways to address things like gullies. Some more technical fixes with technology or structures called gabion baskets, but also importantly with different kinds of plants, trees and grasses, looking at slow growing, deep roots, fast growing, shallow kind of net-like root systems, and using these together to safeguard against future gully erosion. They're a real menace. One of my colleagues, after we left there, he kept saying, I'm still thinking about those gullies. A Zimbabwean colleague of mine, still thinking about these gullies. So anyway, I still think about these gullies too. There are different types of soils, right? We've got agricultural soils, range lands for grazing. We also have wetlands. So I'm going to tell you that this used to be a wetland. Maybe you can't tell. There are districts where lynx is being carried out that are all quite dry for most of the year. have been experiencing higher than average temperatures, erratic rainfall patterns, all the things that we anticipate happening with climate change. People there are living with those realities already. And so what do you do when there's not a lot of water and you need to grow crops? You find the place with the water, right? And so in these wetland habitats, there are natural springs. Wetlands are areas where the water table is quite close to the surface and not flowing over the ground. Great place to plant crops. And so you can see on the top right there, and the top and the bottom right, people have fenced in different springs and wet areas. So one of the goals of LINK's implementing partners is to help families find other ways to access water for their gardens through digging boreholes and these sorts of things. So that people can sustainably move outside of wetlands without increasing their the human capital required to grow food through finding other sources of water. So allowing gardening activities to move outside of wetlands, still be successful, and then hopefully the hope is to restore those wetlands. And so then part of our monitoring, here you can see another one. So you can see some water flowing out on the left there, and then on the top right you can see someone's garden fenced in. So part of what we're doing is also monitoring wetlands to see how well they're doing. Wetlands, of course, are connected to the land through runoff over soil, and we're hoping to see some rejuvenation of wetlands as well. Wetlands do all sorts of things for us. They purify water. They protect against flash flooding. They store a lot of carbon. They're very productive. They have all sorts of materials that you don't find other places that are also useful and of interest to people. I'll just give you one more example of a nature-based solution. Maybe you can see in the inset there in the front this tiny little seedling. This is a plant called, I wrote it on there, Fiderbia albida. And this is a neat tree because it partners with microorganisms in its roots, and it fixes nitrogen. So it can use atmospheric nitrogen and make it usable for plants. And we entered some of these groves where people were growing maize and other things, and it felt, the pictures don't quite capture it, but it felt kind of surreal and very, it was very exciting to see. I think that's what I've got there. Yeah, so LINKS is one short project. It's one aspect of this kind of work happening in another part of the world. Some ways really different from our context here in Winnipeg. But we've seen already these echoes of the sorts of things that soils need to be healthy and to support us and to support other life. And is also what connects us through air and soil to other parts of the world. Right? We're connected with the same desires as people have elsewhere to work with nature to allow healthy ecological cycles that are vital for us humans and also all the creatures that we share this planet with.
Okay, so while John is getting situated here, we are beginning to run tight on time. So I'm going to hit you with one super quick technical question for a term that's going to come up, I think, a few times and already has. Could you just define cover crop for us here today?
Yeah. So it's typically not something that you would harvest, but it's something that can be planted at the end of a season. So here would we use? I'm from BC. We would use something like winter rye out there. You might use clover. It's not something that you're going to harvest at the end of it, typically, but might turn back into the soil in the next season. Or, I think there are different ways to do it, though. I think Heather has better definitions than I'm giving you right now. But yeah, it's this idea that not everything that you plant necessarily you will harvest and monetize or eat. but that the actual covering of the soil in itself is a good that we try to do.
Perfect, thank you.
Good evening. My wife Ron and I on our way here were commenting that it hasn't rained much at all this spring, but it is now, and that means that we all came here and smelled, I think, that springtime smell of soil, right? If you just take one moment to close your eyes and inhale and just think about smelling that smell, we'll come back to that in a second. I want to sketch a quick story for you about a very small scale project from the introductory microbiology course that I taught here at CMU last year, in the winter of 2023. And the labs for that course were fully focused on a sort of semester-long project of isolating and characterizing soil bacteria, in which the students were part of a global search for new types of antibiotics. You're probably aware of the antibiotic crisis that we currently face worldwide. The antibiotics like penicillin and others that we have relied on to treat bacterial infections for decades are losing their effectiveness as bacteria evolve resistance to them. In Canada, we now estimate that about 1 in 4 bacterial infections are caused by bacteria that are resistant to one or more, or sometimes even all, antibiotics. And this is part of a global trend of rising sickness and mortality from infections that used to be much more treatable. So, what are antibiotics? And I just want to clarify, generally speaking, antibiotics are small molecules, and I want you to not confuse them with other things that sound similar, like antibodies, which are very different. Antibiotics are small molecules that selectively kill bacteria, rather than the people or the animals that we're trying to treat. That's a very, important characteristic in an antibiotic, is that it not destroy the person you're trying to treat. They generally don't have an effect on microbes other than bacteria, like fungi or viruses. And antibiotics kill bacteria by interfering with other molecules or processes that are unique to bacteria. And I tried to come up with an example that didn't sound too horribly genocidal, an analogy if you like. So kind of like if you disrupted the production of Jeannie's cakes, you would disproportionately affect Winnipeggers, right? Because that's a Winnipeg-specific thing. Antibiotics specifically affect bacteria because they target processes in bacteria that are specific to bacteria. Anyway, so there's a connection to soil here. And it's because most of the antibiotics that are in use today were originally discovered as products of soil microbes, and in particular of bacteria themselves. Now here I am ignoring fungi, which I ought not to do, but I'm going to. Because most of the antibiotics in use today were actually derived specifically from soil bacteria. So this diagram groups different categories of antibiotics by their original source. And the 2 circles on the left include antibiotics of bacterial origin. And on the far left, the largest circle is for one group of bacteria alone, a fairly small but important group of soil bacteria called the actinomycetes. And when you smell that soil smell in the spring, that's mostly the volatile compounds that are being produced by those actinomycetes in the soil. Just a little bit of trivia for you. The reasons why we have such an abundance of antibiotic production by soil bacteria is that, as we've heard from Heather and from Rachel, soils are highly complex ecosystems. With countless different species, I'm going to focus on the microbes, countless different species of microbes living together and interacting with one another, cooperating with one another, competing with one another, and yes, also suppressing or even destroying one another. And the way that bacteria interact, they don't have language and they don't have fun limbs that they can wave around like this. They interact via the chemicals that they secrete. And so antibiotics are kind of part of those interspecies negotiations. So, to help address the antibiotic crisis and some other issues about how we teach science that I'm happy to talk more about later, Dr. Joe Handelsman, who is a soil scientist and a microbiologist at the University of Wisconsin, developed a project that she calls Tiny Earth. And the goals of Tiny Earth are fourfold. They are to educate students about the antibiotic crisis, to educate students about the soil crisis, because we're also losing good soils at a disturbing rate, to engage students in original scientific research, and last but not least, to discover new antibiotics. That last goal is a particular focus. You've probably all also heard of crowdsourcing. Joe often refers to Tiny Earth as a project of student sourcing antibiotic discovery. So, Joe and her colleagues have now grown Tiny Earth into a worldwide network involving hundreds of post-secondary instructors, thousands of students. They are convinced that many potentially useful antibiotics remain to be discovered from the dirt in our own backyards. And I agree, so I've become a partner instructor in this program and brought CMU into the Tiny Earth collaboration. So what we did last year was to take a soil sample from CMU's backyard, the woods just south of the residence buildings here. Doing that in January at minus 25 degrees was a challenge. And so it was enough of a challenge that I didn't require the students to come along with me, but I should have. It was a missed pedagogical opportunity to get cold and dirty at the same time. Anyway, here you can see the general area, the location on the map, the hole that I dug with the help of some warm water, and a little bit of soil from our sample, which you can see is really typical of this area. It's really clay rich. That's why you can practically mold it into anything you want. So in the end, we spent a semester working with bacteria from a total of 1.8 grams, about a quarter of a teaspoon of soil, from that tiny nondescript bit of earth just a few hundred meters away. By our estimates, it contained about 35 million culturable bacteria per gram, plus an unknown number more, maybe even as much as 100 times more, that can't be grown in the lab. And that great unknown group of soil organisms is part of what makes a project like this humbling, especially to someone like me who focuses more on aquatic worms and cells and knows just enough about soil to be stupid. So it's also a reminder, like the fantastic puns of Genesis 2, that humans, humus, and humility all share the same Proto-Indo-European root word. So the Tiny Earth workflow goes as follows. We take a bit of soil, we suspend it in water to wash the bacteria off the soil particles, and then we dilute that water many times so that it has just a tiny amount of bacteria per unit of volume. And we spread that over agar, basically a gelled, nutritious broth in a petri dish. And because the bacteria are so dilute, individual bacteria will end up in their own sort of turf on the surface of that agar, settle there and start to proliferate. So one bacterium divides and becomes 2, and 2 divide and become 4, and 4 become 8, 16, 32, and I'll stop until we have a colony. A colony is just a visible heap of bacteria, and to be visible, it would have to have probably a few million bacteria in that one spot. Since the bacteria in a colony all descend from a single cell, they form a pure population of a particular strain, or what we call an isolate. So we can use those colonies as a source of cells for further growth and testing of those isolates. And so here's just one dish I actually cheated a little bit, and these are bacteria from soil just by the pond over here that I took before the course, but same sort of thing. With the course itself. So students take those bacteria from the dilution plate colonies and swab them in little patches on fresh plates to make what we call isolate libraries. In total, the 17 students in the class patched over 400 bacterial isolates, and then they patched those bacterial isolates from the library plates onto another set of what we call screening plates, where each plate has a lawn or a film of a tester strain of bacteria. The tester bacteria are harmless relatives of pathogenic strains that have been prioritized or identified as agents of concern by the World Health Organization. So the library isolates are growing on top of a lawn of the tester strain. And if a library isolate produces a substance that kills or inhibits the growth of the tester strain, you'll get a little bacteria-free clearing zone growing around that, that patch of the library isolate. And so that way, we identify isolates that are potential sources of novel antibiotics. We can grow them as pure cultures and work to identify and characterize them. So the students that year ended up sort of focusing their attention on one strain each. So we looked at 17 strains of interest for preliminary study. And this past fall, I had 3 independent study students working and extending that work. And together we've identified 9 distinct strains or species of bacteria that are producing something that has antibacterial activity. This is just a summary table of the activity of our isolates against 4 different tester strains. You can see that they really vary in the spectrum of different testers that they inhibit. The photo here shows a few replicate screening plates. You can see the zones of clearing around several of our soil isolates. So we'll be adding information about these isolates to a central database that's hosted at the University of Wisconsin, describing isolates of interest from different soils and also aquatic sediments and some other interesting environments, literally all around the world. While I was training to become a partner instructor, they were running a virtual training program with a group of partner instructors from Nepal and Pakistan. So it really is global, all continents. The 9 strains that we found may or may not ultimately be medically useful. But the project as a whole is almost certain to bear some fruit, and we'll repeat this process here in subsequent years. In the end, though, we come back to the soil. So in addition to the microbiological techniques and skills that students learn in introductory microbiology, I want my students to appreciate a few other bigger picture lessons. And when I do this in the future, I want to emphasize all of these a little bit more. One of them is that healthy soil smells good. Second, small things matter bigly. Third, we are barely scratching the surface of understanding the complex web of interactions among soil microbes. And fourth, and relatedly, even if we exhaustively cataloged and described all the life in the soil, that would only be a small step toward knowing how to live wisely on and in and with it. A dozen new antibiotics may help for a time, but unless and until we use them more responsibly, they won't solve the issues of human ignorance, pride, impatience, and short-sightedness that really are at the root of the antibiotic crisis. All of this is why it's also important for budding scientists to learn a thing or two about the academic, social, cultural, philosophical, and theological traditions that have been saying these things to us for millennia. Thanks very much.
All right, so I've got one question for you here, and then we'll transition to our last bit of the evening, which I will explain in just a second. But I was listening to an audiobook by John Green recently, his Anthropocene Reviewed. If you're looking for something to read or listen to, highly recommend. And he discusses how the global supply of penicillin arose from a moldy cantaloupe in Peoria. And I was listening to that earlier this week and hearing what you were saying. Could you just reflect on like what it feels like for you as a scientist to know that those discoveries are kind of around the corner and to be looking in soil specifically for you as a creative, curious kind of person?
Well, I mean, that's kind of the way science, and I think research, not just scientific research, but you're always exploring the unknown, and you don't necessarily know where it's going to take you. And that's one of the things I mentioned, the pedagogical importance of the Tiny Earth Project, which is really what Joe would probably want me to talk more about. But when you put students in a situation where they're working in a lab, working at a project where we genuinely do not know where it's going to head, right? rotten cantaloupe is going to produce something that might be interesting. That just opens up a whole different way of thinking about science than students typically get, certainly than I got in my undergraduate training at a university that will not be named.
Well, thank you so much, John. All right. So kind of two components we're going to get to now, the first of which is a pair of responses kind of specifically to some of the things that Heather shared with us. And then once we've done those responses, I'll ask you to step up to that mic and throw whatever you have at these lovely folks to discuss a little bit further. So our two respondents here, reflecting specifically again on what Heather has shared, are Megan Klassen-Wiebe, who, if you are familiar with the geography of this space, you may know of the Metanoia Farmers Worker Cooperative. And Megan has been with them since its beginnings over a decade ago. She has worked with Heather in that space on soil health. And during the winter, it's worth noting that Megan also works at Camp Assiniboia. Our second respondent will be Scott Gerbrandt, the Manitoba director of Rocha, a Christian environmental organization that positions care for one another and the planet as a profound act of love. His connection here is that his home front yard in Wolseley is both awesome and designed by Heather. So Megan, come, and if you give us your best 2 to 3 minutes, I'd love to hear what you have to say.
So I wasn't quite sure where to start. I didn't know what kind of audience we'd have and how many of you would know the farm already. So I'm going to give you a really brief history of what we've been and what we've done. So we started in 2011 as the CMU farm. We tilled up, you can see in the top photo there, just about 3/4 of an acre. And then once it's fenced, it's just under an acre. To plant and grow a CSA, which is a community shared agriculture model business, as a worker cooperative. So we grow vegetables here in the city and sell them to people who eat them. And inherent in our values of the farm has always been care for the land. And we have used some mulches and things in the past among our various practices, but let's see. So you can kind of see what the farm has looked like in other years. We have a lot of rows of things, and some years are greener than others. I think the 2014 picture is actually a spring photo, so it looks a little bit more bare because things aren't growing yet. But in 2020 and 2021, we had a really couple of dry seasons, very drought conditions, and we really noticed that our soils were not well protected. And so in 2023, we connected with Heather to design us a permaculture plan, which looks like that. So some of the things that has changed, or that will change, are the ways that we shape our beds and set up the farm, the ways that we think about water flow on the farm, and the ways that we think about perennial plants and weeds. And so in this first year of permaculture work here on campus, we focused mostly on the permanent beds, building up a no-till system, and working a lot with mulches. And it's been really exciting to see the benefits that we've been able to see just in the first year. We already have noticed that we're not having to water as much, even in a season that's a little bit drier. We're seeing less weed pressure in different places, and when there is weed pressure, we're finding it easier to pull those out because that soil structure is not as compacted as it was before. So we're really excited to see the different ways that that's going to change, and that's going to benefit the soils on the land that we care for.
So these images here are of my front yard. We've been talking a lot about backyards, but I live in Wolseley on a reverse pie shape, and my backyard actually is only 9 feet in the back. I can barely park my car in the back of my house. So this has caused me to do things in my front yard. And similarly to Megan, my wife and I engaged Heather and Paul and got them to design a permaculture plan that for our front yard, well, for our whole yard actually, but it was most of the works are in the front. And I would say our front yard has always been a place of spectacle because we have our eldest son always does some weird, strange things like forging things out of steel in our front yard, so people come and gather. But we have reclaimed our front yard in this permaculture plan. And what you see right here is kind of the initial stages of us enacting the plan. This is in 2022. That first picture you see was just mostly just grass. And just bare, and then we started doing the lasagna garden model where we put down cardboard and mulch, as we've been talking about, and some compost before we're doing some of these plantings. And the other thing that you don't quite see in this image, I didn't quite give, is you'll see in that middle image there, you see there's like that one line of mulch you see right there that is actually hiding something. It's called an urban swale. The two things that really Heather got us to think about right away is not just about how are we taking care of our soil, but how are we managing our water on this site and where is it going and how can we capture that so that we can keep on building that health of that soil. I would say the third thing was then us figuring out what is the right mix of plants that we wanted to put into the ground. And most of it are native plants that we've put into that section, but it's been certainly has been a mix. And the last thing that I would say that I've really learned and have been responding from Heather and Paul's work has been a lasting kind of comment of, okay, Scott, here is the plan. Now you're going to have to do it and respond to it as things change. So we are now 2 years in and we're already noticing, okay, this was really working, but we have to listen to what is not working and maybe make some changes. So really still staying active in terms of our paying attention to what's going on in this place. So we've really enjoyed that work. We still feel very new and novice when it comes to doing this kind of work, but we're certainly enjoying it and have loved it. So yeah, I think my parting shot that I give to you, I know this whole thing has been kind of housed in do something in your backyard, but my encouragement to you is if you've got any soil in your front yard, do something with it or nurture it. Because if one thing we've learned as it relates to things like climate change is unless you're talking about it, we can't really keep on taking a collective action. And if there's one thing I can say that has happened as we have engaged this permaculture plan to our front yard, is I've had more conversations with my neighbors than ever before, because we're actually out in the front yard and we're doing strange things. And they're going, what are you doing? And it has resulted in great conversations. And yeah, I'd encourage you to do that in your front yard, not just your backyard.
Hide it under a bushel? No. So with that, I think we'll turn this to you, our audience. We've got, as allotted, about 11 minutes, but I can be flexible if necessary. If you have a question for someone here or kind of a prompt for us, I encourage you to stand up to the mic. Keep it short. I will wield my power to cut you off if I feel it necessary, but I promise to build a conversation out of your thoughts. So with that, I turn it over to you. If you sit there and look at me, I've got 90 questions here to work with. So fire away.
Okay. We have a front lawn, and I like my lawn, right? Now, we don't use chemicals, so I'm out in the front lawn pulling weeds all summer, right? And I want to keep the lawn. So what is your advice to a guy like me who likes his lawn, wants to keep his lawn in the front? What advice do you have for me?
So as someone that is actively building a native prairie pasture out of my lawn, I won't get on a pedestal and talk here, but I think, Heather, you've got some experience in terms of kind of what that system looks like. So yeah, feel free to weigh in.
Sure. One thing I can say about urban lots is pretty well across the board, you will see compaction, which means anaerobic conditions, which means your soil life is not thriving, which means your plants are not thriving. And likely you've got a bacterial environment, meaning the growing environment favors weeds. So a few things you can do is aeration, right? To get the air into the soils, to shift away from anaerobic microorganisms. In anaerobic conditions, there are microorganisms at work, just not the ones that we want and that will benefit our plants. And so aeration, and then building your soil health through organic matter. So if you feel comfortable, leave your leaf litter to actually feed your lawn. Like that's the action of a tree dropping its leaves, right? Is it feeds the soil biology. So aeration, feed the soil with organic matter where you can. And then there are actually companies, I don't think in Manitoba, because this idea of microbiology inoculation is really new to Manitoba, but there are companies, lawn companies, that are servicing residents and et cetera, to restore the microbiology in lawn contexts. So there are ways, just first steps, aeration and feed your soil life, so that you can shift away from that bacterial growing condition.
Feel free to step up. I think if anybody's undergoing a raking in fall responsibility debate within your household, what I'm hearing is you can resolve that and bring about positive change for your yard.
Thank you, all three of you, four of you. Heather, you mentioned a good compost and bad compost, but you never defined what constitutes bad or good.
Okay, so if we look at the history of the composting industry, I think it's probably safe to say that the purpose of the composting industry isn't to create good compost that will feed your soil biology, but rather as a waste management strategy. So in that sense, it often happens that compost is created in anaerobic conditions, which favors anaerobic microbiology, which is not what you want for your soil and for your plants. So things to watch for: if it smells like vomit or pee, those are signs of the bacteria that you don't want. Soil is supposed to smell nice, right? If it smells like a forest floor, you've got a better compost. It'd be nice if you had a microscope. Then you could actually analyze the hypothetically. Then you could analyze the microbiology and actually see the biology that's there. But those are some signs. So if it smells putrid or rancid or whatever, that's a sign of compost to stay away from. If it smells earthy, that's a really good, a good sign. Is that helpful? Yeah.
So Evan, can you inspire us on the prairie front grass approach?
I spent a few summers working at Prairie Originals, which is a native plant greenhouse north of Winnipeg, and I did it for a few reasons, primarily to earn money so I could fuel my studies financially. And my parents anticipated that in borrowing their vehicle and driving 25 minutes north of the city, I would return home with financial independence, which I did, but I also came back with what we called the sick bay plants, those which were pots containing several species that couldn't be sold as one, those that for the faint of heart would not be sold and worth $8. But to me, these were treasures that needed to be saved and given a new home. And so my mom discovered me in the backyard with a spade in hand, installing a small prairie garden in her backyard. And it worked great. I have, at one point, close to 40 species of Manitoba native plants going on in my mom's backyard. And whereas we have now negotiated a more tolerable collective space around how that's supposed to work, there are still native plants growing in my parents' backyard. And I think that taught me a few things about, I think, relationships, but it also taught me how the power of beauty in that space is really important, and giving these plants a chance to speak their language and to be discovered and to be shared is a really powerful tool. And so I went on that journey with my mom in discerning kind of, this discovery together. And now I think what I've done is I've decided, and this has been a decision I had, in my mind for a long time, was that if I ever bought a house, I would want my front yard to be a space to showcase this. And I don't think, Sheila, I'm not the only one on Beaverbrook that has plants doing that work. When we purchased our place, we went for a walk nearby, and I spotted that I wasn't the only one growing those plants in the neighborhood. And so if I can make a sales pitch for this idea, because it's not lawn grass, which has its function, I've got somewhere in the neighborhood of 11 crocus flowers almost blooming right now. And I've been thinking for weeks about how this is a fireworks show about to go off in my front yard. And I think if you can instill that excitement in other people, if you can share those colors and those smells and those spaces with other people, you can start conversations that, like you mentioned, help begin this reimagining what our spaces can look like and what our relationship with the natural world can look like. It's been an awful long time since those plants bloomed in River Heights, but there are 2 yards where that's happening now, and I'm optimistic that if we all do this, eventually these conversations get bigger. So, Prairie Originals opens in like 3 weeks. Head out to Selkirk, come back with some plants, plant them in someone else's yard. Okay, so, failing another question. Rachel, you mentioned that carbon cycles up into the atmosphere, but some carbon is retained and sequestered in the ground. Can you talk about how it's cycling, but also not totally cycling, so some sticks around?
Yeah, that's good you picked up on that, because it was real oversimplification, wasn't it? Because the key thing in soil health, or one of the key things, is that there's also organic matter in it, right? And that's why we add mulch and we add compost, because we want to build up that organic matter in the soil. And that's also why part of why healthy soils are important for fighting climate change. They contain a lot of carbon just in their biomass, right? Most living and dead organic matter is carbon that, when it's decomposed, ends up as carbon dioxide in the atmosphere, or methane under the worst case scenarios of anaerobic decomposition. But also those healthy soils with all that organic matter allows plant roots to go deeper and to be more robust, and that's where we get a lot of carbon storage. And then we get the fungal hyphal networks that we learned about as well. And so, in healthy soils we see oxygen-based decomposition returning carbon to the atmosphere, but also a lot of storage that then later feeds that food web and feeds plants.
All right, John. I know that you've discovered some microbes, or at least revealed some to the naked eye in the neighborhood here. But can you give us a sense of how much variation exists? So if you took a bunch of soil samples here, how different is that to my place not too far from here as compared to Winkler or beyond?
I have no idea.
Awesome.
And this is what I mean by just how humbling this is. And I've talked a little bit with Megan about, we should go and see, how the soil at the farm differs from the soil out here on the lawn and that sort of thing. And that kind of project, if you want to sample in a way that you're actually capturing, some of the real diversity of things. Because there are billions of bacteria per gram of soil, it becomes an enormous project really quickly, which is just beyond my capacity as a non-soil microbiologist. But a couple of things that other people I think have found, Rachel talked about how different the sort of soil invertebrate communities were between two places that were really quite close together, and I suspect you would find similar kinds of patterns in soil as well. One thing that's interesting is that studies that people have done in sort of large scale, like geographical studies of the diversity of soil microbes, is that we're used to thinking about like there's more diversity in the tropics usually and less towards the poles, right? That's true for plants and animals. And more diversity in sort of wet places, like again, think of wetlands or tropical rainforests, and less in drier places. But it turns out that soil microbes really kind of buck all those trends. And that the diversity of plant life actually doesn't correlate particularly closely at all with soil microbial diversity, and that some of the most diverse micro ecosystems are in places like the arid Southwest in the continental U.S., and that there's not this sort of latitudinal gradient either. That Arctic soils are as microbially diverse as tropical soils.
I can imagine that, for me at least, hearing the penicillin story kind of knocked me on my feet a little bit. Can you describe what it looks like when, for any of you really, but John, I guess specifically here, when you see students encountering those kinds of surprising trends? Is there a moment that stands out to you in terms of watching a student's eyes light up in that space, or like realizing kind of how these systems work, or take us by surprise?
Yeah. Like students comment on this is really cool. And the other thing that I think starts to happen is that they start to develop, it's almost like anything where the more time you spend with something, the more you start to appreciate it, even if it's something that you might have thought was boring in the past. And like bacteria don't have a whole lot of personality, but they do form these colonies with amazing shapes and colors, and so like just aesthetically, I think students get into it as well. Like the flip side of it is that you spend a lot of time doing things that don't really seem all that exciting, right? Like a lot of science is just washing dishes. And so that's one thing that I've had comments on. Like it just didn't feel like we were really moving very quickly, right? Because we all have this sort of CSI vision of science as you go and you put something in a machine and it spits out the answer for you. And the answers are usually kind of ambiguous and take some effort to decipher. So I definitely see that spark. But it also comes with, and maybe it's partly caused by some frustration along the way as well.
When I saw those images, the first thing that struck me was not their beauty, but the wave of anxiety that still lives with me when I think about my agar plates in the microbiology lab and just hoping that those streaks were in the right little pattern. But yes, also very, very beautiful. Come on in.
My name's Ray Vanderzeg. I teach international development studies. When I worked in Haiti 30 almost years ago, working with poor farmers, working the land was sort of the lowest on the totem pole. That's what you did if you couldn't get a job in the city or in town or become a teacher or do something else. Yet all of you today have told stories about how you love the dirt. I'm not going to say this without getting emotional. My dad was a farmer and he loved the dirt, right? And so I think the key to all of these things is learning, and this is what you've done today, is you've taught us a little bit how to love the dirt, love the soil. And so can you just say a little bit more about that? Because it seems to me that's the thing that binds the stories together, but it's also the key to getting us to pay better attention to the soil and what the soil can give us. Because in our society, I was going to say capitalism, and I'm not against capitalism, but the soil doesn't get paid well because it's free. And we overuse things that are free. It's the classic public goods problem, right? So how do you get, and so the solution to that is to get us to love the soil more so that we're willing to pay a little bit more for our groceries and to take care of our front lawns differently and do those other things. So just say a little bit more about helping us, and you've done it already, so I'm thanking you for it as well, but just putting it in that way, it seems to me that's what you're telling us to do, is love the soil more.
Right, Heather? Help us fall in love.
Okay. Well, if I think of my own journey, I spoke about this journey through despair, and which I think is a common story at this point, right? We're facing certain challenges, and I think it has to do, for me, it has largely had to do with how I understood my own place, how I understood my own role on Earth. And I think, growing up, we learned, okay, if you shut off the lights and shut off the tap when you're done brushing your teeth, we'll be fine. The polar bears are going to be fine. And of course, the issues are so much more systemic than that and so much bigger than that. Taking it, I would say, an environmentalist perspective still put me at odds with the earth, because no matter what, I was always a draw on the earth. My presence means damage to the earth. Innately, humans are a draw. And when I encountered permaculture, something switched, something flipped, where I was able to suddenly see that, wait a second, stewardship doesn't, maybe I can think about stewardship differently. Maybe we're actually meant, maybe we say this on our website, humans were made for the earth like walnuts were made for maple ice cream. We're meant for each other. We're not just part of the earth. We are the earth. We are that humus, right? That is us. There's not actually that much of a divide between us and the soil. The same microbiology that's in the soil is, there it is again. The same microbiology that's in the soil is in our gut, right? So if the soil is healthy and we're eating food from a healthy soil, we're feeding our guts with healthy microbiology. So it's hard to find the divide between us and the soil. So permaculture allowed me to shift my thinking that maybe humans are meant to be a benefit. Maybe we have a role in helping the earth sing. And then I discovered this soil food web concept. And it was just like it, permaculture helped certain things click for me, and then it was like the soil food web concept grounded it. Like, oh my goodness, here is an example of where I can quite literally have an immediate impact on the soil and help the earth sing. And so for me, it's about learning about my proximity to the soil, that there's really no separation between my body physically and the soil, and then learning the role of the soil in determining whether our world, the plant world, the human world, thrives or dies. And so that's where my love of the soil has come to see just how who was it that said, like, we know so little of, yeah, John, you were saying, there's so little we know about the soil. All of the people that I have encountered who have been studying the soil for decades still say, we're just, like, this is just the tip of the iceberg. And so there's so much more to learn about the role of the soil in the thriving or the demise of the world above ground. So I don't know if that answers your question.
Rachel, when you hear that idea of like that connectedness, what's your pitch from, I guess, a community perspective and kind of blending, abandoning perhaps nature-based solutions and thinking about community embeddedness as a love story?
Yeah, one thing that struck me as we were visiting farms and seeing the stark contrast in biodiversity was that what your father felt about the soil is universal, is international. Let's say international. I don't know if there's soil on other planets, but it's global, right? The places that we tend, they care back in a way, and they bring forth life. Wilson coined this beautiful term, biophilia. We have this, this innate love for other living things, and the soil is sort of the epitome of of life, right? It's where life springs on land, and it's full of life itself. And once you get to know it, then it's hard to not respond with that feeling.
All right, John. Closing pitch.
I was going to say we, and here I can very much say I. I'm not very good at learning to value the things that are really important, or I value them, but I'm not really good at finding validation in the truly important stuff, right? Which often is the very humble things, and so I like. I don't know where to start, but I think about the experience I had as an undergraduate student, having just maligned my undergraduate education. One of the best things that I ever got to do, was made to do, was to go make an insect collection for an entomology course that I was taking, right? And insects are definitely among the category of things that we don't give a whole lot of attention to. But the way the world came alive for me as I started to pay attention to where those, because I'm looking for insects, I want to find things, I want to find a diversity of things, and the world just comes alive. It's just a different way of framing things, and that was really wonderful. So, yeah.
I think that we are about at the end of our time here. So thank you for sharing your pitches. I want to echo briefly what John says there. If an insect collection feels inaccessible to you, I am delighted to announce that it is April. And April is bird migration beginnings. So if you feel like you want to begin to discover those connections, this is your moment to look both down as the flowers begin to bloom and up as the songs come back. I have it on good authority that the ruby and golden-crowned kinglets are back, folks. They're going to be angry. They're going to be puffing up their spiky little colorful heads. This is your time. I heard sandhill cranes overhead. It's like a frog croaking hundreds of feet above your head all day, every day for like the next week. It's awesome. This is your season. As I was listening to some of the things being shared here, I had to pull up my book quotes page here and to share one more thing from J.B. McKinnon, who, if you've heard the 100-Mile Diet, that's him and his partner writing. I encourage you to look up what he's written. And the thing that stuck with me and my, I guess, hope for you is that you would take this to heart. And to reflect on this. As a question of individual and collective values, it is perfectly legitimate to like nature, read soil, to want more of it and to stake your position on grounds of curiosity, mystery, and delight. All those things are evident in what we've heard today, and I encourage you to go forth with those things as you look up, as you look down, and you experience the world around us. So with that, I want to say one more thank you to these folks for sharing with us. Put your hands together for our panel today. I hope that you have enjoyed connecting here today. I know that I certainly have. Take care as you go, and thank you for getting into the weeds with us tonight.